The Lamb of God. Idea strand 20. Kings open double doors, double barriers, double gates, double ram gate. Bacteriologists know that polar localization of proteins is critical for cellular processes such as division, chromosome partitioning, and cell cycle control in E. coli. Physical differences exist at the cell poles, such as large polar caps or gaps between the cytoplasm and cell wall and protein enrichment sites. Also, the concentration of secretion and adhesion machinery to the cell pole may facilitate host invasions, such as the phage lambda, which attaches to the Lamb receptor site of the E. coli. Entry into the Earth cell or the E. coli bacterium is through its double cell membrane or double barrier at the phage lambda receptor site, the Lamb porin. Porins are water-filled pores or channels across the outer cell membranes of the gram-negative bacteria, such as E. coli. Maltoporin, or the Lamb porin, is responsible for guided diffusion of maltose and maltodextrins for E. coli, along with being the phage lambda receptor site. Three-dimensional studies of Lamb show that each subunit of the trimetric protein contains a wide channel formed by 18-stranded anti-parallel beta barrel. Three inwardly folded loops, an umbrella structure, are located in the channel lining, along with the greasy slide, a hydrophobic path composed of aromatic residues. In the drawings in the Amduat and Book of Gates, the activity takes place along a three-channel pathway that may represent the three channels of the Lamb porin. Lamb, a guided diffusion channel, is one of 6 proteins composing the maltose transport system in E. coli. Bacteria favor the carbon source of glucose, and maltose is a dimer of glucose. However, in its absence, bacteria will access lactose. Researchers have identified a mutant of the maltose transport system that transports lactose efficiently. The mutant has a missense mutation in the MAL-F gene that changes leucine to tryptophan. Mutations allowing lactose transport have a lac plus phenotype. Scientists theorize that the maltose transport system may contain 2 figurative paths of the transport of substrate. In one path, the substrate is bound to the MBP, the maltose binding protein, in the periplasm. In the case of lactose transport, the mutant accesses only the second low affinity path. Since MBP cannot bind the substrate, lactose. The Book of Two Ways also references these 2 figurative pathways. On the macroscopic level, these 2 paths easily translate to the LLBL and the HLBL of the magnetopause. Lactose transport is by proton symport. 1 proton, 1 lactose are transported by the lactose paramase, a product of LAC-Y gene. Any ion, such as a hydrogen ion, in excess outside of the cell can act as a driving force for the transporting nutrients. Recall that the polar cusp is in the excess of the solar hydrogen ions. The sun god represents not only a composite of hydrogen ions and other energetic particles, but also the incoming phase lambda that will ferry the deceased into the bacteria host for transformation. We have posited that the genetic heritage of the deceased inserts into the incoming phage lambda at the CI gene, inactivating it. This is called insertional inactivation. Isis is also present on the sun god's supramolecular bulk and may be the representation of lactose and its resultant transport through the cell by the hydrogen driving force. For if the lambda vector is carrying the LAC-Z gene, it may be active, as represented by Isis, Lady of Provisions, on the sunball. LAC-Z, or the B-gal gene, is the enzyme normally synthesized by E. coli cell when lactose becomes available. LAC-Z may be present in the vector as it is for the lambda insertion vector, Charon 16A. However, DNA inserted into LAC-Z inactivates the gene just as it inactivates the CI gene in lambda. It is interesting that the lambda prefix still sits silently at the B-gal gene, which is the LAC-Z, in the E. coli host chromosome. When the prefix excises, it can sometimes leave the part of this gene, a type of the specialized transduction, activating the LAC gene by leaving the seat empty. Related to this idea is the symbol of the name of Isis in Egyptian, which is the empty seat or throne, suggesting that an empty seat or throne indicates the gene seat active for lactose metabolism, instead of inactive and occupied by the prefix in the lysogen. Thus, an empty gene seat could signify the excitation of the prefix at the resurrection of Osiris. So decoded, we have Egyptian polar gap or dockyard of gods equals lamb porin. Polar bursty reconnection site equal injection of recombinant lambda DNA. Isis's name, empty throne, equals empty seat due to prefix excitation. As we expand our microcosmic model of the sun god operating like a lambda vector, the earth representing an E. coli host bacterium, and the deceased functioning as a DNA insert, it seems practical for understanding the Egyptian afterlife. In support, we have posited that the union of the deceased and the sun god at the northern polar cusp is similar to the insertion of a exogenous DNA fragment into the incoming lambda genome. The sun god is a ferryman carrying the deceased through the duat in a similar fashion to that of the lambda cloning vector. Entry into the duat or mountain of the West Gap is similar not only to absorption and injection of lambda DNA into the E. coli host at the lamb porin, but also to the injection activity of FTEs into the magnetopause. In addition, polar localization for cell cycle control in E. coli suggests a rationale for the Egyptian advice to go to the polar cusp of Earth, which is enriched with high energy particles. The presence of Isis on the sunbalk at the northern polar cuffs may indicate lactose transport by proton symport. Likewise, insertional inactivation of the CI in the lambda vector may indicate that the LAC-Z gene is functional and not inactive by an insert in the vector, so the lactose enters the bacterial host. Similarly, the excitation of the lambda profit from the LAC-Z gene may represent the meaning of the empty throne designating the name of Isis. All, the voyage of the deceased sun god and crew of 8 traces a figurative path for humans to the next level of evolution, cosmic clone. Objective 6 is completed. The supermolecular nature of the deceased sun god and the crew of 8 supports the idea of an afterlife cloning experiment that designs the human's evolutionary future if one bonds to the sun god. With this detailed examination of phage lambda, its land receptor site on E. coli, and the potential of maltose transport systems for lactose passage, we can easily decode additional Egyptian signs in the interest of finding out how the deceased sun god restores the Eye of Horus. However, it will be helpful for the reader to refer to Figure 9.2 as an aid for understanding scientific ideas and chemical processes. Okay, the rest of the page is real fast, y'all. Got a screenshot. Definitely not holding it there. Okay. Alright, right on, y'all. Next is chapter 10. We see you soon.